All right, so today we're going to look at paper 6, which is the alternative to the practical for biology, and that's IGCSE Cambridge International Examination, CIE, May-June 2017, variant 1, so 0610/61. All right, let's get started.
We are on in 7, 6, 5, 4, 3, 2.
Question 1. Proteins are an important part of the diet. These proteins are used in the body to make enzymes and other cell structures. You are going to estimate the concentration of protein in two solutions, A and B, using the results from a set of standard protein solutions. Known concentrations of protein solution were made. Using Burette reagent, the color intensity score was determined at each concentration. Step 1. 7 test tubes were labeled 1, 2, 3, 4, 5, 6, and 7. Step 2. Solutions containing different concentrations of protein were made using the volumes of 1% protein solution and distilled water shown in Table 1.1. Okay, this is Table 1.1. We have the volume of 1% protein solution that was included. So test tube 1 didn't have any protein, and test tube 7 had 4 centimeters cubed of 1% protein solution. And then we had the volume of distilled water, and test tube 1 had 5 centimeters cubed, and test tube 7 had 1 centimeter cubed, so in total there's 5 centimeters cubed of solution in all test tubes. And then they calculated the percentage concentration of protein solution. So question A1. Complete table 1.1 by calculating the percentage concentration of the protein solutions in test tubes 3 and 7. Write your answers in table 1.1 and show your working. So to calculate the percent protein in 3, take percent protein in 3, in test tube 3, that equals the amount of protein solution. So that is 0.50 centimeters cubed divided by the total amount of solution, that was 5 centimeters cubed, 5.00 centimeters cubed. And then to get percentage, you multiply it by 100. However, the thing you have to look out for here is it's 1% protein solution. So now we have to take this answer and multiply it by 0.01 or 1%, which is exactly the same as dividing by 100, multiplying by 100 and then dividing by 100. So those kind of cancel out. So the answer you get here is 0.10%. All right. Okay, so that is 0.10. And if you look at that, it's 0.10 lies between 0.05 and 0.2, so it's likely the correct answer. Okay, so percent of protein in 7 is the same procedure. 4.00 centimeters cubed divided by 5.00 centimeters cubed times 100 times 0.01, so it cancels them both out, and that equals 0.80%. 0.80. So first of all, you'll notice that there's no percent symbol in here. You will lose marks because it says percentage. It doesn't have the symbol here, but it's the same thing because they've written percentage. And it also, I put 0.10, 0.80, and that's because all the other numbers have 2 decimal places. Okay, so you just try to stay consistent. All right, next. 2. State the color that shows the presence of protein when tested with Burette reagent. Okay, that is lilac. You could also say, purple or mauve or violet. Okay, those are pretty much lilac. Okay, but lilac isn't the word they like to hear. It's I don't know, they just think it sounds nicer, I guess. Step 3, so that was step 1 and 2, calculating the percentage concentration of protein solution. Step 3, they took 2 centimeters cubed of Burette reagent and it was added to each of the solutions in test tubes 1 to 7. Each test tube was shaken gently to mix the contents. Step 4, the test tubes were placed in a test tube rack in order of concentration, from the least concentrated to the most concentrated. Step 5, 2 centimeters cubed of burette reagent was added to the protein solution in the test tube labeled A and shaken gently to mix the contents. And step 5 was repeated for the protein solution in test tube B. They figured out what the color change would be for 7 known concentrations, and now they're testing 2 unknowns, and they're going to compare it against the known ones. Step 7. Test tubes 1 to 7 were held against a white background so that the color of the solutions were clearly visible. These test tubes are shown in figure 1.1, and this is figure 1.1. Step 8. The color intensity of the solution in test tube 7 was given a score using table 1.2. Okay, so this is table 1.2, a nice, precise set of pluses. It's not very precise, really, but it works. Okay, it's difficult to put a number to a color, and that's basically what they're trying to do. Okay, so 3. Use table 1.2 to compare figure 1.1 by writing in the score for test tubes 1 to 6. Test tubes that have the same color intensity should be given the same score. Okay, so the fourth, test tube 7 is given for you, and that's the darkest, and that has 4 pluses. Test tube 1 is the lightest, so that should have 1 plus. And then in between, you just judge where it looks like they change color slightly. Okay, so these two look about the same, and they look darker than this one. So plus, 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 plus. Okay, this one looks slightly darker. Plus, plus, plus. And this one also is darker. Plus, 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 plus. Okay, there's a little bit of leeway they'll give you. If you judge this one to be the same as this one, or maybe judge these two to be the same, there's a little bit of leeway they'll give you. But for the most part, palest on the left, darkest on the right, and using this plus scale. Okay, and 4, prepare a table to record the results for test tubes 1 to 7. Your table should show the concentration of the protein solutions, the color intensity score given to each solution. Okay, they haven't really given us much space here. Normally they give you too much space, and you have enough to work with, so you just have to write small. Okay, but remember, in biology you need to draw in all the cells. Okay, so first of all, we have in the left-hand column, we should write in the concentration of protein solution in percent. Okay, and you could write percentage concentration of protein solution in the same way they did in the first table, or you could write concentration of protein solution with a line with percent. Make sure these are clear enough. And in the next column, you should write color intensity score and say a little bit what the score is. Okay, so I put this in brackets because it's not really a unit. It's just a bit of a description about it. Okay, I'm just going to write in the numbers here. So this is 0.00, 0.05, 0.10, 0.20, 0.40, 0.60, and 0.80. Now I will draw in the cells, and then I'll fill in the color intensity score afterwards. Okay. Okay, so remember, when you draw in cells, you have to make sure that all the lines are drawn with a ruler. Okay, so the color intensity score at 0, was very pale, and then it went to 2 pluses, 2 pluses, then we had 3 pluses. We're just writing down what we put in above. 3 pluses, then we had 4, and then we had 4. Okay, good. The results for A and B are shown in figure 1.2. So 5, use figure 1.1 to determine the color intensity scores for test tubes A and B in figure 1.2. Now this is easier to do when you have the paper right in front of you, because you can put them right next to each other. But if we go up to figure 1.1, okay, the first test tube, it could be anywhere, one of these 3 really. I'm going to say it looks closest to 3. And the second test tube is very dark, so it could be 6 or 7. They pretty much look the same really anyway. So we can call that with 4 pluses. Okay, so I'm going to say plus, plus. You could do 3 pluses there. And this one is plus, 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 plus. Pretty much has to be 4. It's the darkest one. All right. Okay, 6. Use your table from A4 to estimate the percentage concentration of the protein solutions in test tubes A and B. Okay, it's an estimate. Okay, so if we have a plus plus, you could say it could be 0.05 or 0.10. You could have put down 3 pluses and you could say it's 0.20. It doesn't look like the 0.401 though. So you could put in any number between here. I'm going to write down 0.10. Okay. So that's 0.10%. But it's a range. It could be anywhere between 0.05 and 0.20. You can't say 0.20 though if you only put 2 pluses up here. Or you can't say 0.10 if you put 3 pluses up here. For test tube B, you had 4 pluses. So let's look at the table. So really, looking at this table, with 4 pluses, you could say 0.60 or 0.80. If you'd only put down 3 pluses, you could say 0.40. So it depends on what you put down. Okay, I'm going to say 0.40. All right, that's good. B1. Identify the control for this experiment and explain why it is used. Okay, remember, the control is not the controlled variable. They're different. The controlled variables, there's many of them. Those are the things you're keeping the same. The control is basically the test tube or the experimental situation where you expect nothing to happen, or the negative, where you have something to compare against. Because you do the experiment with all the ones with burette in, but you want to know what happens when there's nothing in it. Okay, so the control in this one is test tube 1 that only had water. And the reason why it is used is to show the color it would change when there was no protein present. So the control is test tube 1, which only had water, and the purpose is to show the color when there is no protein present. All right, good. 2. Explain why the method used to find the concentration of the protein solutions in test tubes A and B can only be an estimate. Okay, the key here is that it is a qualitative measurement. It's not quantitative. There's no real number on it. You're just saying it's around this number. It's darker or it's lighter, rather than it's a 1 or a 2. You assigned numbers, you assign those little pluses, but they're not really proper numbers. So it is a qualitative measurement. You're looking at a quality. Is it darker? Is it lighter? Rather than a number. Okay? And because you're just trying to judge a color, you're looking at it by eye saying, oh, that's kind of darker than that. It means it is a subjective measurement. So it is a qualitative measurement, so it is subjective. You can also say it is judged by your eye rather than a specific way to measure it. So that's another way of saying it's subjective. Okay, another reason why it can't be accurate is because similar concentrations look the same. So you can't judge between ones that look the same. So it is a qualitative measurement, so it is subjective, and similar concentrations look the same. Okay, that's 3 possible marks out of 2. So if you missed one out, that's okay. Question 2. Yeast cells have a cell wall on the outside that appears as a dark layer and a large paler colored vacuole occupying most of the cell. The nucleus is round and often near the center of the cell. It is much smaller and darker than the vacuole. Yeast grows by forming small buds on one side of the cell that eventually break off. Figure 2.1 shows a photograph of yeast cells viewed through a microscope. A1. On figure 2.1, label 2 of these features of yeast cells. So the features they're talking about are the cell wall, the vacuole, the nucleus, and the buds. Okay, so those are the only features you should label. You shouldn't try to identify anything else. Okay, let's look at the diagram. Okay, so if we look at this, it's easy to say, oh, the dark thing in the middle has to be the nucleus, but it's not, because if you notice, this is smaller and this is pretty big. This is taking up most of the cell. That structure there is round and it's taking up most of the cell. Sometimes it's small, but sometimes it's bigger. So this is the vacuole. Okay, the nucleus is actually really quite hard to see because there's also little granules in yeast cells which at this level look very similar to the nucleus. So I would avoid labeling the nucleus. You only have 2 things you need to label, so I would avoid labeling the nucleus. Okay, the cell wall goes around the outside. That's that darker, outer line. Okay, and the bud, if you notice, this one has a little bud coming off. This one has a little bud coming off. This one has a slightly bigger bud coming off. This one has a bud coming off. So the buds are just the parts that are breaking off. Okay, so we have to label 2 of those. I'll try and label each of them, but you only need to label 2, and you only should label 2. Label the 2 you're most confident with. So probably the easiest one to label is the bud. And remember, that's a straight line with a ruler. There is no arrowhead at the end. You just put it towards the actual structure. Okay, so this is the bud, the smaller bit coming off of the bigger bit. Okay, another thing that should be nice and easy to label is the cell wall. You can draw that from any cell. It doesn't have to be the same one. So that's the cell wall. And if you notice, I am not writing on the photograph. I'm writing outside of the picture. You should always do your labels so they're outside of the picture. They even when they did it, cell A and B are done outside of the picture. P and Q are label lines. That are not label lines. They're not labels. They are measurement points. So you don't need. They don't follow the same rules. But you always draw your label lines with one straight line, and you make sure that the lines don't cross. If you drew the cell wall as a line across the bud line, that would be wrong. They might not take a mark off it for it at this level, but they might. So you don't want to risk it. Okay, and so the next thing we're going to draw is a vacuole. So let's draw a nice big vacuole. Here's a nice big vacuole. Okay, and then let's see if we can find a nice nucleus. Okay, there's a little dot right here. I'm going to draw that as a nucleus. Okay, but now that I've drawn the line, you can't even see it. So I would probably not draw the line to the nucleus. I would have drawn it to the things that are much easier to see. Okay, because you only need to draw 2 of them. You don't draw them all. All right, let's move on. 2. Measure the length of 4 yeast cells and show where you have measured each of these cells on figure 2.1. Record the length of each cell and calculate the average length for all 4 cells and include the units. So we write down the length of the 4 cells here, but we have to make sure we draw it on the photograph. Okay, so there's a little bit of give when it comes to these measurements, because the examiner probably won't measure all 4 cells, but they might. They actually might. So I'm going to measure this one. And so you draw a line. It goes from the top to the bottom, and you do that little marks to show that it's the end. And this one I measured as, I measured that as 11 millimeters. Okay. I'm also going to draw a line for this one. And again, you draw the marks at the end to make sure that you can show where the beginning and end is. And I measured that one to be 10 millimeters. Okay. I also drew the line, I also measured this one. And I measured that one to be 11 millimeters. Okay, and finally, I measured this one here, and I measured that one to be 9 millimeters. Okay, it doesn't matter which cells you measure, but you do have to draw these little measuring lines. Okay, so now we record those down below. So we have 11 millimeters, 10 millimeters, 11 millimeters, and 9 millimeters. You might measure them slightly differently if you've printed yours off on a slightly different size paper to what I did. That's okay. Okay, Now we need to take the average length of the 4 cells. So if we take 11 millimeters plus 10 millimeters plus 11 millimeters plus 9 millimeters, we divide that all by 4, we get 10.25 millimeters. But if you notice, all of these are measured to the nearest millimeter because it can't really measure very easily to the half millimeter or quarter millimeter, so we could do it as 10 millimeters. Okay. 3. Measure the length of cell A along the line PQ, drawn on figure 2.1, and include the units. Okay. Cell A, I measured that using my ruler to be 11 millimeters. Okay. But the mark scheme says it is 12 millimeters. And that's because I printed mine out on a slightly different scale to what is given out in the exams. But that's okay. It actually says 12 millimeters plus or minus a millimeter. Okay, so that you have even a little bit of leeway there. Okay, so I'm just going to use 11 millimeters for my calculation later on, but you could easily use 12 when you're practicing. So measure the length of cell A. We measured it as 11 millimeters. Okay, so calculate the actual length of cell A using the formula. Actual length equals the length of the drawing over your magnification, and give your answer to 3 decimal places and include the unit. Okay, this is again a part that people often forget. For some reason, it's easy to kind of miss this last line in a question. Happens all the time. Okay, 3 decimal places. Okay, so the actual equals 11 millimeters divided by 800. And the reason why it's 800, if we look at this drawing up here, it says magnification times 800. So it's 800 times magnified. So that equals 0.01375 millimeters. That's clearly too many units, as it says to 3 decimal places. So we take equals 0.014 millimeters. We round up. Okay, 0.014 millimeters. Okay, next. All right, my least favorite part of any biology exam, the part where you need to make a drawing, because I'm not an artist. But the good news is you don't need to be an artist. Actually, quite often artists are the ones that get the lowest marks on these questions, because they like to add some artistic flair, maybe shade things in, or try to add in every single detail. Okay, so remember, when you're doing a drawing in biology, simple, smooth lines. Okay, if that's the drawing. Okay, you do not draw where the lines don't touch. I know that's an exaggeration, but you need to make sure if you draw a circle, that's a bad circle. You lose marks for that. You need to make sure a circle, that's a terrible circle. Make sure your circle touches. Remember, you also don't feather. You don't shade. You don't stipple. Stippling is a way of doing shading where you're just putting little dots to show that there's a texture there. You don't do that. All you do is you're basically drawing the outlines. Okay, and the examiner knows you're a biology student, you're not an art student. You have to do your best to draw things in the same orientation, to the same approximate size and shape. But if you don't make it 100%, so every little bump and lump is exactly the same, they're okay with that as long as the majority of your diagram is okay. Okay, so you're drawing this what's in this box. So this line here, and you're drawing this line here, and you can see this very clearly. You can see this very clearly. You can see 2 little circles here very clearly. Okay, the other thing you probably notice is this outer line is thick. So if you drew it, it's not as thin as, this is a really thin line. This outer line is slightly thick. So if you drew it with 2 layers, that would probably be good. In this particular instance, they don't take a mark off for that, but quite often when the lines are this thick, they will take a mark off if you don't do 2 lines. Okay, so I'm going to draw it with 2 lines even though they don't take a mark off this time. Okay? All right, let's draw this yeast. All right, so this is my drawing of the cells in the box. Okay, it's not perfect. There's a little bit of wobble on it, more than I'd do on a piece of paper. I drew in 2 lines here to show that there's a thickness to the cell wall. This particular time, there aren't marks for or not for having the cell wall thick enough, showing that it's not one-dimensional. But quite often there is, because it is thicker. Okay? Things you have to show, things you get marks for. You get marks for it, the lines all being one smooth, continuous line and not having any shading or stippling or feathering. Okay, you're not supposed to shade in the slightly darker sections in here. You need to make sure that it's taking up at least half the space given to you. And you need to make sure that you could show the main structures you can see. So you can see 1 big circle in this one and 2 smaller circles in here and 1 big circle here. And another mark is for these cells, having the 2 cells with the one with the bud coming off of it, and that they're in the same, as close as you can get, shape, orientation, so facing the same way, and position. Okay, so making sure that you've done your best to actually draw what's in the cell, what's in the box, rather than something that looks similar. Okay, this is not a great diagram, but that would probably get you the full marks. Moving on from the drawing question. B. Some students measured respiration in yeast using a culture of active yeast. Yeast produces a gas during respiration. 2 syringes were filled with 20 centimeters cubed of active yeast culture, and each syringe was placed into a large test tube containing water at 35 degrees Celsius. Both were placed into a water bath at 35 degrees Celsius as shown in figure 2.2. The volume of gas in each syringe was measured every 5 minutes for 25 minutes. So the gas produced by the yeast collects at the top, and this is the yeast, the active yeast. The water bath is 35 degrees Celsius, and here's the test tube that the syringe is placed into. And as the gas is produced, the plunger goes up, but also some of the yeast will probably come down. Okay, 1. State 2 variables that have been kept constant in this method. Okay, there's about 4 of them that you could easily put down, but you only put down 2 because it has 2 listed. Okay, so one of the variables that was kept constant is the starting volume of the yeast. Okay, the starting volume of the yeast was 20 centimeters cubed for both of them. Another thing that was kept constant was they were both in a water bath at 35 degrees Celsius. So the temperature of the water bath was kept to 35 degrees Celsius. Other things you could have put down, that the time intervals for measurement was kept at 5 minutes for each, and the total measuring time was 25 minutes for both of them. Okay, those are all valid variables that have been kept constant. 2. Identify one source of error in this method and then suggest an improvement. Okay, there's lots of things you could have put down. Okay, and don't forget, for here, an improvement means an improvement for this exact error, not an improvement for something else. So they have to match. Okay, and remember, whenever you're talking about sources of error, human error does not get you a mark. If you say, well, they read the scale wrong or something like that, they did it wrong, that's not an error because that was a mistake. That is not something that you can improve next time other than with practice. And that's why you generally do more than one and you take an average. Okay, so let's start listing some of these. You could say that as the reaction occurred, there was yeast lost from the bottom of the syringes. So if you look at this diagram, yeast came out the bottom, and when the yeast came out, that's a lot of the respiration that was occurring because that yeast would be respiring, and a lot of the gas that should be collected wasn't being collected. Okay, so that's a source of error. Okay, so yeast was lost from the syringe, so there was less respiration and gas collected. And the improvement is to seal the syringe. You could also use a gas syringe method. So that is collecting it above. Okay, putting it into a test tube and then attaching a gas syringe, which is a different setup. Okay, so some other things that you could have written down. You could have said that when they collected the yeast, they might have taken in some air or some froth with the yeast, because if you've ever done an experiment with yeast, you'll notice that there's a lot of froth on the top. So the way to improve on that is to make sure that they fill the syringe from below the level of foam. A third thing they could put down is that the yeast samples may vary in concentration. So if they take one sample of yeast and then they set it up and then they take another sample, they may have taken it from slightly different levels in the culture, and there may be more yeast at the bottom than at the top. So what they should do is they should stir it before taking the sample each time. Okay, another method they could use is that there was no method of maintaining the temperature. Basically, they put it into a beaker of water at 35 degrees Celsius and expected it to stay at 35 degrees Celsius for 25 minutes. That's not going to happen. So either they should put it in a thermostatically controlled water bath, they should heat it with a Bunsen burner with a thermometer in it, or they can just put insulation around it. So it will be more likely to stay at that same temperature. Okay? When they put the syringes in to the water bath, they didn't give any time for the yeast to reach 35 degrees Celsius. They measured it immediately. So that means they didn't give time to equilibrate the yeast before using it. So the way to improve on that is to leave the yeast for a little bit of time so it can reach the same temperature of 35 degrees Celsius. And another thing that could be a possible source of error is that these syringes have quite large differences for the graduations. Okay, so basically the way to improve on that, so it's a very imprecise scale. So the way to improve on that is to use a syringe with more graduations. Okay, so any of those would be good. Make sure that the improvement matches the error, and then you'll be fine, but only put down one because it only wants one. C, table 2.1 shows the results. So the results are listed in this table. 1, calculate the average volume for gas collected at 25 minutes. Okay, so gas collected at 25 minutes, 13 plus 14 divided by 2 equals 13.5. 13.5, and you don't need to put any units because it's in the title. All right, and then 2, plot a graph on the grid of the average volume of gas collected against time, and you need to add a line of best fit. Okay, so for this one, so you need average volume against time. So time is what is changing, and average volume is what is measuring. So time goes on the x-axis. Time, and that's in minutes. All right. And the y-axis is the average volume of gas collected in centimeters cubed. All right. So looking at the scale, we have to go from 0 to 13.5 or 14 or 15 for the y-axis. So let's do the y-axis. So the easiest way to do this is actually a bit more tricky, because if you go up by ones, you don't have enough. If you go up by twos, you only use half the graph, and you want to make sure you use as much of the graph as possible. So the easiest way to do this is you go from 0. Every large square is 2.5, 5, 7.5, 10, 12.5, and we go up here, that's 15. Okay, so if you look at that, each 10 squares is 2.5, so 2.5 divided by 10, so it means that every single square is 0.25 per square. Okay, so every square is 0.25. All right, so on the x-axis, that's a bit easier. So we have to go from 0 to 25. That's easier because we could just go 0, 5, 10, 15, 20, 25. And if you're using up less than half the graph, you know you've done it wrong, and so you need to change the scale. Okay, so I'll just plot the points. Okay, so these are the points that you're given. Okay, so you could draw your line of best fit. Looking at this, it probably is actually just a straight line. And at 0 minutes, there should be no gas collected, so you can start from 0, 0. Now what I'm going to do is just draw a straight line. That's kind of the average of all of these points. Okay, and if you look at that, it doesn't really go through any point, but that's okay. It's the average of all of these points. You don't really want to draw a dot to dot. Okay, you want to do the best you can to draw a straight line. You could try drawing a curve like this as well, freehand. But if you draw a curve, you don't want to use a ruler. Okay, one or the other. All right, good. 3. One of the students decided that the result collected in syringe 1 at 15 minutes was anomalous. Suggest a reason for the decision. Okay, so syringe 1 at 15 minutes. Okay, so 2, 5, 7, 12, 3, 7, 11, 13. The difference between 7 and 11, that's a pretty big number compared to 5 and 7, 2 and 3, and 12 and 13. Okay, and 13 and 14. So the reason why the student thought that it was anomalous is because the difference between 7 and 11 is pretty big compared to all the other results. So there is a large difference between syringes 1 and 2 at 15 minutes. And that's 1 mark's worth. All right, so finally, question D. Describe how the students could use the method in 2B to investigate the effect of pH on respiration in yeast. Okay, so this is an experimental design question. There's always an experimental design question. And experiments, you need an independent variable. You need a dependent variable. You need some controls. You need maybe you want to modify the method. You don't have to modify the method, but you can modify the method. You should discuss reliability. When I say discuss reliability, repeat twice. There you go. That's reliability. Okay. You might want to discuss safety. You might want to discuss the control. Okay, so these are things you can discuss, you can put down. And this question is out of 6 marks. So that means you can't really complete every single part of this. So you need to make sure you have some points from as many of these as you can. Okay, but make sure for the controls you don't list your entire list in the entire method of saying the things you're controlling, because you generally only get 2 or 3 marks for listing the controls down. Okay, so we're going to start with the independent variable. Okay, you're measuring the difference in the pH. So that means you need to change the pH. Okay, so the first thing you could write down, keep in mind you can always use different wording. Take 3 or 5. 5 is a really good number. 3 is the minimum, 5 is better. Take 3 20 centimeter cube portions of yeast culture and add 1 pH solution to each to make them different pHs. Okay, so test the pH of each culture to make sure they are correct. You could say test the pH with litmus paper to make sure they are correct. You can and choose 3 different pHs. pH 7 is a good one because that's neutral. A little bit acid and a little bit alkali is good. You can do 5 different numbers as well. You're not actually doing the experiment. It doesn't matter if it would be more difficult. Okay, now we're going to discuss the dependent variable. Okay, so you're going to measure the volume of gas given off every 5 minutes for 25 minutes because you're modifying the method in 2B. So that was the method that they used there. Okay, now we're going to put down one or two controlled variables. Okay, so use a thermostatically controlled water bath at 35 degrees Celsius. That's one of those key words that they like, the thermostatically controlled water bath. That just means it's an electronic water bath, not you using a Bunsen burner with a thermometer in, which also works, but the thermostatically controlled one can be kept a little bit more accurate. So add the same volume of pH solution to each culture. Okay, same volume, that's a control. Okay, now we're going to describe a slight change to the method. So use a gas syringe to collect the gas. Okay, that's a slightly more accurate method of collecting the gas than the one that was described. You don't have to include this step if you want to just use it the same method. We can also discuss reliability. Okay, so use 3 syringes of each pH and take the average. Okay, that's doing it 3 times per pH. All right, and finally we can talk about safety. Okay, so you can say use tongs to remove syringes from the water bath. However, it was only at 35 degrees Celsius. It's probably not that dangerous. Or you could say to wear goggles as different pHs are used. You'll only ever really get 1 mark for safety, but that's a mark. Okay, so reading through this entire answer, you take 3 20 centimeter cube portions of yeast cultures and add 1 pH to each to make them different pHs. Okay, test the pH of each culture to make sure that they are correct. pH 5, pH 7, pH 9. Measure the volume of gas given off every 5 minutes for 25 minutes. I didn't mention that you could put each test tube into a water bath and attach the gas syringe in that, but you can put that in there. Okay, use a thermostatically controlled water bath at 35 degrees Celsius. Add the same volume of pH to each culture. Use a gas syringe to collect the gas. Use 3 syringes of each pH and take the average. Use tongs to remove syringes from the water bath or wear goggles as different pHs used. Only put down one of those because you'd only get 1 mark. Okay, now if you notice, that is a lot more than 6 points. You don't need every single one of those. You just need enough to get 6 marks plus a few extra in case not every one counts as a mark, because sometimes only 1 or 2 from a particular section will count. All right, that is the end of this exam. If you've enjoyed this, if you found it useful, please give me a thumbs up so that I know, and please subscribe so you can get some more. And if you have any other questions, comments, or anything else you'd like to say, please put them in the discussion below and we'll respond as soon as we can. And have a great day.